Welcome to the fascinating world of single-cell protein, or SCP. Proteins are essential to life, but what if we could produce them not from animals or crops, but from microorganisms? Did you know that tiny microorganisms can serve as food for humans and animals? But before we understand SCP, let's first look at what proteins really are. Proteins are biomolecules made of amino acids responsible for growth, repair, immunity, and energy supply. They are often called the building blocks of life. They are essential for muscles, enzymes, hormones, and antibodies. Without sufficient protein, our body weakens. In our body, proteins build and repair our muscles, tissues, and organs. Proteins can also act as enzymes, hormones, and antibodies. They are important for our body metabolism and in transport of gases, like hemoglobin carrying oxygen. Moreover, they are an important part of regulation of body function, for instance, hormones which control several key metabolism of body. Apart from regulatory function, they also provide energy when needed. On average, an adult requires 50 to 60 grams of protein per day. But due to increase in world populations, billions of people worldwide struggle to meet this requirement due to food scarcity. Traditional protein sources are not enough to meet the protein's requirement of this huge populations. This is why scientists, biotechnologists are looking for alternative way to meet the huge demand of proteins. And what if various microbes can serve as rich source of proteins? This idea sounds excellent. To meet the rising protein demand, scientists explored alternatives to conventional food like meat, milk, and pulses. That's where single-cell protein comes in. Microbes that can be grown rapidly and converted into edible protein, offering a sustainable and affordable solution. The idea of eating microbes is not new. In the 19th century, yeast was first used as a protein source. Thus, SCP is part of our diet since ancient times. During World War I and II, yeast and algae were tested to feed soldiers during food shortages. In the 1960s-70s, major research programs explored bacteria and fungi. In 1950s, British Petroleum initiated production of SCP on commercial basis. Protein, 72% protein content, was the first commercial SCP used as animal feed. The name single-cell protein, SCP, was introduced by Professor Scrimshaw of MIT in 1967. Protein was produced from Bacterium Methylophilus methylotrophus that was cultured on substrate methanol. In 1985, a breakthrough came with mycoprotein from Fusarium venenatum, commercialized as corn. The mycoprotein, marketed as corn, went through a 10-year evaluation process before being approved for human consumption in 1985. Today, algae like spirulina and chlorella are popular as superfoods. Selection of microorganisms for production of SCP is a very important step. Not every microbe is suitable for SCP. The beauty of SCP lies in diversity of microorganisms that can be cultivated for its production. Therefore, SCP-producing microbes should meet some characteristics features, such as to be useful for SCP production. First, microbes must contain around 40 to 80% protein content, meaning high protein content is preferred. Second feature is SCP-producing microbes must grow faster with less time and should have high productivity rate. Third feature, SCP-producing microbes must have ability to grow on cheap substrates such as food waster, agriculture waste, industrial waste, etc. On the other hand, SCP should be safe, non-toxic, safe nature, and digestible. Last but not least, SCP should have balanced amino acids composition for best nutrition, meaning SCP should have all essential and non-essential amino acids. There are mainly 4 different types of microbes used in SCP production. These include yeasts, such as Saccharomyces cerevisiae, Candida utilis, fungi, such as Fusarium venenatum, SCP product called corn, algae, such as spirulina, chlorella, and lactobacteria, such as Methylophilus methylotrophus. Yeasts are widely used because they grow rapidly and contain 40 to 50% protein. Examples: Saccharomyces cerevisiae, baker's yeast, Candida utilis. These yeasts can be grown on feedstocks such as molasses from sugar industry, whey from dairy industry, sulfite, waste liquor from paper industry. The SCP produced from yeast is used in baking, brewing, flavor enhancers, food supplements, animal feed. Fungi can yield up to 60% protein with good amino acid balance. The commonly used fungi used as a Fusarium venenatum, from which a commercial SCP product corn is popular as meat substitute. These fungi can be grown on starch waste, agricultural residues, corn steep liquor. Interestingly, the SCP produced from fungi has a meat-like texture, therefore accepted as vegetarian protein. The SCP produced from fungi such as corn is sold in 15+ countries as burger patties, nuggets, and sausages. Algae are rich in 60 to 70% protein, plus vitamins, minerals, and essential fatty acid. Commonly used algae include Spirulina platensis and Chlorella vulgaris. These algae can be grown in ponds, lakes using sunlight, CO2, and simple nutrients. The SCP produced from algae are good health supplements of essential nutrients, fortified foods, animal and fish feed. Spirulina tablets and powders are widely available as superfoods. One of the advantages of using bacteria for SCP production is that they multiply extremely fast and can contain up to 80% protein. For example, Methylophilus methylotrophus produced bacterial SCP from methanol. Many low-cost substrates such as methane, methanol, agricultural and food industry waste can serve as feedstock for bacteria and other microbes to grow and produce single-cell protein. But bacterial SCP also has some limitations, such as the microbial biomass often has high nucleic acid content, which can be harmful if consumed directly, so it must be reduced before use. Processing and safety checks are needed to make SCP suitable for food and feed applications. In short, SCP is a promising, sustainable protein source but requires proper processing to overcome its limitations. The process of SCP production includes preparation of substrates, like molasses, waste biomass, or even natural gas, inoculation with selected microbes, large-scale fermentation in bioreactors, harvesting microbial biomass, processing to reduce nucleic acid content, drying and packaging. Today, several SCP products are already on the market. Corn, a meat substitute. Spirulina and chlorella supplements sold in capsules and powders. Yeast extracts, used in baking, flavor enhancers, and animal feed. Bacterial protein meal, explored for animal nutrition. SCP finds applications in animal feed, poultry, fish, cattle, human food supplements, protein powders, fortified foods, biotechnology, enzymes, bioproducts. SCP offers several benefits. Very high protein yield compared to crops. Short production cycle, independent of climate. Can utilize agricultural and industrial waste. Rich in amino acids, vitamins, and minerals. Can help reduce global malnutrition. However, there are also limitations. High nucleic acid content, which must be reduced. Possible allergenic or toxic compounds. High processing cost. Consumer acceptance issues. Taste and texture differences compared to natural food. With advances in biotechnology, SCP could become a key part of the circular bioeconomy. Innovations like genetic engineering, waste-to-protein biorefineries, and cheaper fermentation systems will make SCP more accessible. In the future, microbes may become as common on our plate as traditional foods. In conclusion, proteins are essential for our body, and single-cell protein offers a sustainable way to meet this demand. From history to modern applications, SCP is not just science fiction, it is already shaping the future of food and nutrition. Thanks for watching. Please like, subscribe, and share. And